Hi there, my name's Lucas, and the title of our project is Moving Fast and Slow, exploring the relationship between node of Ranvier length and axon conduction regulation in nerve cells. This work was conducted by Arancibia Carcoma et al., and the title of their work was Node of Ranvier Length as a Potential Regulator of Myelinated Axon Conduction Speed.
This study conducted research on a physiological mechanism of one of the most complex systems in the body, the nervous system. The nervous system controls everything from growth and development to our thought processes, senses, and movement. In order to do this, the system must transmit information in the form of electrical signals across nerve cells, and this information is then processed to produce the appropriate response. An insulating layer of protein and fatty substances called myelin are used to transmit information in an efficient manner. Myelination is therefore vital for healthy central nervous system functioning, and it allows for brain processing to be significantly faster.
So, how do nerve cells transmit information via electrical signals? Particular areas of the axons of neurons, which are nodes of Ranvier, facilitate this process. It is known that nodes of Ranvier are regions that are of myelin that aren't covered, which allows for the influx of positive ions and therefore depolarization of the membrane in these regions. Depolarization of the membrane can then contribute to the generation of a fast electrical impulse. Although it is not well understood how the length or frequency of nodes of Ranvier are determined in neurons, which could be a key process for regulating speed of conduction.
The study was conducted specifically to investigate whether neurons can actively change the length of the nodes of Ranvier to modulate conduction speed. The researchers hypothesized that altering node length could allow for the body to meet varying information processing needs. And this stems from the notion that myelin thickness adjustments may also be used to tune conduction speeds. MRIs of humans and rodent cellular studies suggest that myelination can actively increase when learning motor tasks, such as piano playing, juggling, reaching, or running. There were three main steps the researchers used to test their hypothesis. First, they measured node length and axon diameter in rat optic and cortex nerves to determine how much variation naturally occurred. They also carried out sodium channel measurements to find if a relationship existed between both channel density and node length. Then, using fluorescent dye injections, they analyzed whether variability in node length mainly occurred along the axons or between the separate axons. And lastly, the authors conducted some mathematical simulations of nerve impulses to elucidate the effects of node length alteration on conduction speed. If one was to assume that adjustment of node length can tune conduction speed, then all node lengths along the same axon should have similar or equal lengths. In other words, variability should be greater between different axons than on the same individual axon. And so to test this idea, fluorescent dye was iontophoretically injected into the cortex. Node length distribution along single axons was much narrower than that of all axons, and you can see this is visualized in figure 2D, which shows that the mean coefficient of variation for node length along individual axons was 48.8% lower than that of all axons measured. And as such, node lengths were confirmed to be similar along axons but different between axons, which further implies that these axons may tune conduction speed by adjusting node length on an individual basis rather than a collective one.
So, as mentioned prior, the authors used measurements derived to conduct simulations in order to examine the effect of node length alteration on conduction speed. In short, a model was used in a computational software to conduct simulations on hypothetical axons that were divided into three parts: the node, the paranode, and the intranode. A number of assumptions and parameters were defined for varying scenarios, and within time steps and current flow across the axon membrane was calculated using the parameters shown here in the slide. This would then help us better understand how conduction speed varied based off the node of Ranvier properties.
Data measured prior in the study suggested a positive correlation between sodium channel number and node length, which implies a fixed channel density. However, node lengths could also vary independently, which implies the exact opposite, that channel density is not fixed across different nodes. The authors therefore required a compromise, and so they modeled two extreme situations. One where either channel density was held constant, or another where channel number per node was held constant and density of channels varied inversely with node length. It should be noted that two main factors will affect conduction speed: nodal capacitance and internal exon resistance. An increase of either will decrease the conduction speed. So, when the number of channels is held constant at each node, numbers here, there is a negative correlation between conduction speed and node length, as indicated by the straight dashed line. This occurs because an increase in nodal length increases nodal capacitance, as well as inner resistance to current flow. However, when nodal ion channel density is held constant, a third factor comes into play. As node length increases, the number of ion channels will also increase, leading to an increase in conduction speed. This is shown in the same plots, so you can see where the solid curve peaks at a maximum. Past this peak, increasing node length decreases speed for the same reasons as mentioned prior. Differences in plot A and B result from cortical axons having nodes around 1.5-fold longer, as well as a 1.7-fold shorter internode length. So the nodal membrane would contribute a larger fraction of the total membrane capacitance, and therefore varying nodal length produces a larger range of conduction speeds.
Next, the authors assessed how powerful node length changes were in tuning conduction speed as opposed to altering the myelin sheath thickness. So, some changes were observed that were made for decreasing conduction speed. First, looking at the myelin sheath thickness number, when it decreased by one sheath thickness, we would see a decrease in conduction speed by about 34% in the cortex. Sodium channel density decreases and internode length increases could also yield the same speed changes in the cortex. In other words, to produce equivalent speed effects, the change in membrane area needed when tuning node length versus myelin wrap thickness is 1,000% fold less in the optic nerve and over 200-fold less in the cortex. This means that it is more efficient to actually change the nodal length and may be done more rapidly compared to myelin sheath alteration.
The grand conclusion here is that tuning conduction speed by altering node length is much more efficient than altering myelination, especially when considering lipid and protein metabolism that is required with membrane alterations. This occurs because minor changes in nodal length can produce significant changes in conduction speed. Now, as discussed earlier, the relationship between node length and sodium channel density suggests that node length may be adjusted through inserting or removing membrane with these channels. However, it may also be possible to vary node length independently of the channels, as modeled in the simulations, by endo or exocytosing vesicles without sodium channels in the membrane. Regardless of this relationship with sodium channels, node length alteration offers an advantage over myelin sheath thickness alteration. Due to its efficiency, large changes in conduction speed can more rapidly be made at the node, allowing for tuning of axons on an individual basis. So, based on these conclusions, we can now understand, for example, that many individuals with abnormal release of glutamate may experience nerve degeneration as a result of less information being gathered due to possessing longer node lengths.
These findings possess implications for treating neurodegenerative disorders. Nodal amyloid precursor protein, which is commonly associated with the onset of Alzheimer's disease, was proposed to be a regulator of node length in a separate study. This suggests that changes to its regular processing may alter node length and thus play a role in its association with Alzheimer's disease. Finding a way to reverse the abnormal amyloid precursor protein function may be a way to treat Alzheimer's disease in the future.